The interaction between mushroom and dead tree is saprophytism, decomposition. Fungi act as decomposers that break down dead wood and organic matter using enzymes, releasing nutrients back into the soil for plant growth. The interaction between bees and a non-flowering tree is commensalism. The bee benefits by using the tree as a resting or shelter site, gaining protection and a safe place to perch. Meanwhile, the tree remains unaffected, as it does not receive any benefit or experience any harm from the bee. The interaction between epiphyte and tree is commensalism. Epiphytic plants grow on the trunks or branches of large trees to gain support and better access to sunlight. The host tree remains unaffected by the presence of the epiphyte.
The interaction between trees growing close together is competition. In freshwater ecosystems, aquatic plants compete for sunlight, space, and nutrients dissolved in water. This competition affects plant distribution and plays an important role in maintaining ecosystem balance. Trees growing close together compete for sunlight, water, and nutrients. This competition influences plant growth and contributes to the vertical structure of the forest.
The interaction between Venus flytrap and dead tree is predation. Venus flytrap is a carnivorous plant that captures insects using modified leaves. After trapping the insect, the plant releases digestive enzymes that break down the insect's body into simpler nutrients. These nutrients, particularly nitrogen, are absorbed by the plant to support growth in nutrient-poor soil, while the insect is harmed and killed.
So here's the list of content. We have Lab Report Practical 1, Basic Concepts of Statistic and Variation. Graph Lab Report Practical 1. Lab Report Practical 2, Factors that affect the distribution and abundance of organisms.
Practical 1. Basic Concepts of Statistics and Variation. In this practical you will learn about normal distribution and its parameters, the mean and variance, and initial understanding about correlations. The objectives of this practical are as follows: to calculate and describe the following descriptive statistics: mean, variance, and standard deviation. To understand the concept of correlation. To determine the relationship between the two variables. Materials: leaves, ruler, weighing balance, graph paper. Method: 1. 10 leaves were taken, measured and recorded their length and width. The means, variance, and standard deviation of the length and the width of the 10 leaves were calculated. The data was tabulated in a graph paper. 2. Another 10 leaves were taken and repeated as above. Repeated this with another 3 sets of leaves. The results were tabulated as a data matrix. 3. 1. The data of all 30 leaves were used to plot a graph of leaves length versus leaves width. 2. Check if there was a relationship between the length and width of the leaves. If there was, discussed what sort of relationship it was. Was it linear?
So here's the result for the leaf length, width and weight in set 1. Then here's the result for the leaf length, width and weight in set 2. And here's the result for the leaf length, width and weight in set 3.
So here's our graph result for Lab Report 1. As you can see, there is y-axis presented the length, and x-axis represented the width.
Move to next, which are discussion and conclusion. For discussion, this experiment highlighted the key concepts of statistics and variation. Descriptive statistics allowed us to summarize and present the data effectively. Measures of central tendency, such as the mean, median, and mode, provided insights into the typical or average values within the dataset. Measures of dispersion, such as the range, variance, and standard deviation, helped us understand the extent of variation present in the data. In conclusion, this experiment aimed to introduce and explore basic concepts of statistics and variation. Understanding variation and statistics enhanced our ability to interpret and utilize the data effectively. And for conclusion, this experiment is success because it able to demonstrate a basic statistics using width and length of the collected leaves to obtain variance and standard deviation.
Next, we have post-lab questions. Question 1. Discuss the differences or similarity of the mean of length between different set of leaves. Do the same for the mean of width. There are differences of the mean of the length between each set. Mean for set 1, set 2 and set 3 are 6.49, 13.44 and 16.4 respectively. The differences between each set because the collected plants are from different species. So, the size and length of the leaves is different and not constant. The leaves from upper part of the plants maybe have smaller size compared to the leaves from lower part because lower part receive more nutrient than upper part as it is more exposed to sunlight. The mean for width for set 1, set 2 and set 3 are 2.93, 6.25 and 5.56 respectively. Set 2 and set 3 have similar mean for the width of the leaves, and mean for set 1 is a bit different, but the difference is not much. This is because the width of the leaves depends on the nutrient absorbed by the plant, whether it is from abiotic or biotic factors. The more nutrient absorbed by the plants, the more growth and development that the plant can undergo. For question 2, what is meant by variance? The leaves have different measurements of width and length. What is the meaning of this statement? Discuss with respect to variance. What is meant by standard deviation? Variance defined as mean squared difference between each data point and the center of distribution measured by mean. There are variation in length and width of a group of leaves. There is no similar individual in population, whether in appearance or size. Meaning of this statement: the leaves have different measurements of width and length. Also about the measurements of width and length have different value on different leaves, due to their different parts of plants which from different parts of stem, upper part, middle part and lower part. So, according to this experiment, there are 10 leaves for each sets, which the total of leaves are 30. From the table, total length is 64.9 centimeters, 134.4 centimeters, 164.0 centimeters. Meanwhile, total width is 29.3 centimeters, 62.5 centimeters and 55.6 centimeters for set 1, 2 and 3 respectively. Furthermore, the value of variance in length for set 1, 2 and 3 is 1.003, 5.07 and 6.28. For value of variance in width is 0.2685, 0.17 and 1.2604 for each set 1, 2 and 3. Based on this result, it shows that there is a different measurement in length and width for each leaves. A standard deviation is a measure of how dispersed the data is in relation to the mean. It is calculated as the square root of the variance. Standard division has divided into two standards, which low standard and high standard. Low standards deviation means data are clustered around the mean, and high standard deviation indicates data are more spread out. And for question 3, why is it necessary to have different sets of measurements for the measurement of leaf width and length? It is necessary to have different sets of measurements for the measurement of leaf width and length because it can record each of the growth to each plant and decide whether it have same characteristics. It also helps to obtain record for the average of the growth in plants.
Practical 2. Factors that affect the distribution and abundance of organisms. The biotic factors of an environment are all organisms found within that environment. A group of organisms of the same species found within the same environment is called a population. Populations of different species sharing the same area and interacting with each other make up a community. One way of studying the interactions of organisms in community is by taking an inventory of all the species in the area and comparing the sizes of their populations. In this practical, you will be exposed with a simple concept of quadrat. Students also need to observe the abiotic factors of a study site and classify the species of plants in that site. The objectives of this practical are as follows: 1. To introduce the simple concept of quadrat. 2. To enhance the power of observation. 2. To record the difference in plant distribution within the quadrat. 4. To infer from natural observations the possible factors that affect distribution and abundance of organisms. Materials: 1. Raffia string. 2. Measurement tape. 3. Stick. 4. Scissor. 5. Notebook. Method 1. In this practical, slopes of different types site at UITM Chawangan Negri Sembilan. Campus Kuala Pila were chosen as the area under the study. 2. One quadrat of 1 meter by 1 meter at each site was made. 3. All the plant types that were found inside the quadrat were observed and the data was recorded in the table. 4. All the data and information then used to estimate the population site and population density within the entire habitat.
So here's our result for Lab Report 2, Quadrat 1.
Here are the post-lab questions. Questions 1. Define the term quadrat in practice. A quadrat is a small, typically rectangular or square area used to sample vegetation or other organisms in ecological studies. In practice, quadrats are employed in various sampling techniques, such as quadrat sampling or plot sampling, to obtain quantitative data about species composition, abundance, and distribution within a defined area, to analyze and compare different ecosystems. Question 2. What are the two basic requirements of all the quadrats techniques? Two basic requirements of all the quadrat techniques are the area of the quadrat, length x width, counted, and the number of organisms of species in the quadrat. Question 3. What would be the advantages and disadvantages of using a larger, smaller quadrat to survey the sedentary organism? If the survey is done on a smaller quadrat, the advantage is less or no chance to miscalculate the species of sedentary organisms, while the disadvantages of smaller quadrat are low frequency of species or miss too many individuals that can result in underrepresentative estimates of population sizes. On the other hand, the advantage of surveying the sedentary organism on a larger quadrat is there will be a variety of sedentary organisms found, which can lead to errors, and the comprehensive survey is not practical. Lastly, the advantage of using smaller quadrat is less time-consuming, while a larger quadrat can be complicated to be carried out and time-consuming to place and sample. Question 4. Problems with spatial scale often make it difficult to directly estimate average densities in habitats such as large forested area or in aquatic habitats where one cannot directly see the animals or plants of interest. Explain the issues concerned ecologist in conducting the sampling method. Since it is usually impossible to sample an entire population within a study area, one must take smaller samples of the community or population of interest. The sampling method needs to have the details about the densities and dispersion patterns of organisms or types of species in the population. Then, the size of the sampling plots, where the sampling plot will be placed, and the numbers of the sampling plots needed to be conducted. And for question 5, why is there difference in the number of plant species in the quadrat? Hypothesize or make your own assumption. Discuss the factors that would have caused this type of distribution. The difference in the number of plant species in the quadrat is caused by a combination of abiotic and biotic factors. Variations in light availability, soil moisture, nutrient levels, and habitat conditions influence which species can grow successfully in the area. At the same time, competition among plant species affects their ability to establish and persist. As a result, dominant species occur in high numbers, while other species are present in lower abundance, leading to differences in plant distribution and species composition within the quadrat.
For discussion, the quadrat sampling method provides a simple and effective approach to study the distribution and abundance of plant species within a given area. Proper selection of the sampling site is important because the placement of the quadrat influences the types and number of species recorded. Through systematic observation within the quadrat, patterns of plant distribution and dominance can be identified. Quadrat sampling allows ecologists to compare species composition and relative abundance within and between habitats. Dong, 2015. The use of a 1 meter x 1 meter quadrat is appropriate for sampling herbaceous plants and other sedentary organisms. This quadrat size balances practicality and accuracy, as it is large enough to represent the plant community while still being manageable during data collection. A quadrat that is too small may underestimate species diversity, whereas a larger quadrat may be time-consuming and difficult to sample accurately. Therefore, selecting an appropriate quadrat size is essential for obtaining reliable ecological data. Archo, 2007, based on the results from Site 1, Front Tree Area, a total of 7 plant species with 114 individual plants were recorded. The species Axanopus compressus show the highest abundance, accounting for 52.63% of the total population. This indicates that Axanopus compressus is the dominant species in this area. Its dominance may be attributed to its rapid growth rate, ability to spread vegetatively, and tolerance to varying environmental conditions. These characteristics allow it to effectively compete for space, light, water, and nutrients, limiting the establishment of other plant species. The second most abundant species, Sporobolus diandrus, 24.56%, further suggests that the environmental conditions at the site favor grass species. Grasses generally grow well in open areas with sufficient sunlight and well-drained soil. Other species such as Trypagandra cerulata and Strobilanthus reptans were present in moderate numbers, indicating that while they can survive in the area, they are less competitive compared to the dominant grasses. Zhu, 2004, species such as Chromalina odorata, Trifolium repens, and Lindonia crustacea showed low abundance. This may be due to unsuitable microhabitat conditions, such as limited light penetration under dominant plants, lower soil nutrient availability, or differences in moisture levels. These abiotic factors, including light intensity, soil texture, moisture content, and nutrient availability, strongly influence plant growth and survival, leading to variation in species distribution within the quadrat. Dawson, 2017. In addition to abiotic factors, biotic factors such as competition play a significant role in shaping plant distribution. Dominant species like Axanopus compressus may suppress the growth of smaller or slower growing plants by occupying most of the available space and resources. This competitive exclusion results in fewer individuals of less competitive species, causing uneven species distribution within the quadrat. Gull, 2019. And to conclude, quadrat sampling does help in understanding and determining factors that affect the distribution and abundance of organisms. By systematically sampling across different microhabitats, we gained a comprehensive understanding of the community composition and structure. From this practical, the distribution and abundance of organisms are influenced by a lot of factors like the abiotic conditions and the biotic interactions. Abiotic factors, including temperature, moisture levels, and light availability, were found to significantly shape the suitability of habitats for various organisms. Biotic interactions, such as competition, predation, and symbiosis, on the other played a pivotal role in determining organismal distribution and abundance. Overall, this practical emphasizes the complex interplay of abiotic and biotic factors in shaping the distribution and abundance of organisms within ecosystems.
Thank you for watching our assignment tasks. Hope you guys doing well, and don't forget to subscribe,
share,
and like.